Thursday, September 14th, 2023, AGNDD Intellireport topic: Tsetse fly. Please pardon any visual and informational inaccuracies in this entirely AI-generated Wikipedia-sourced video article.
Tsetse flies, known for their medical and economic significance, have been extensively studied by entomologists. This text explores the etymology of the word tsetse and highlights its usage in scientific and development contexts. The word tsetse means fly in Swana, a Bantu language of southern Africa. It is a pleonasm, but recently the term tsetse, without fly, has become more common in English, especially in scientific and development contexts. The word is pronounced as Tsetse in Sotho languages and is easily understood in other African languages. During World War II, a British aircraft called the Tsetse mosquito was used for anti-submarine purposes. The tsetse flies have been extensively studied by entomologists due to their medical, veterinary, and economic significance. Their biology is well understood as they can be raised in a laboratory and are relatively large, making them easier to analyze. Tsetse flies can be seen in three forms: larvae, pupae, and adults. The larvae have a maggot-like appearance and quickly develop into pupae, which have a hard external case. Within the puparium, the tsetse completes its development. Finally, it emerges as an adult fly. Adult tsetse flies are relatively large, with distinct characteristics such as large heads, separated eyes, unusual antennae, and a wider abdomen. These characteristics distinguish them from other types of flies. Tsetse flies have a distinct body structure with a head, thorax, and abdomen. The head has large eyes and a proboscis, while the thorax is large and has 3 pairs of legs, wings, and halters. The abdomen is short but wide and can change in size during feeding. Internally, tsetse flies have a large crop for accommodating a blood meal. The reproductive tract of females includes a uterus. Tsetse flies are physically tough and require more effort to kill compared to other flies. The tsetse fly has a unique life cycle that involves adenotrophic viviparity, where the female retains and nourishes one egg at a time within her uterus. The larvae develop internally and are fed by a milky substance secreted by a modified gland. After reaching the third larval stage, the larvae leave the uterus and develop a hard outer shell called the puparial case. During this time, they undergo morphological transformations before emerging as adult flies. The larval stage lasts around 20 to 30 days, and the larvae rely on stored resources and the nutrition provided by the mother fly. The most important hosts for Suidae are overall. The breakdown of blood meals by species is as follows. G. swinnertoni primarily feeds on warthogs and giraffes. G. austini feeds on bush pigs, bovidae, and various duiker. G. fussiplurus feeds on bush pigs, giant forest hogs, and hippopotamuses. G. tabaniformis feeds on red river hogs and porcupines. G. morsitans feeds on warthogs, bovidae, especially kudu, buffalo, bushbuck, and eland, and domestic cattle. G. fusca feeds on bushbucks, red river hogs, and aardvarks. G. brevipalpus feeds on bush pigs, hippopotamuses, and bovidae, especially buffalo and bushbuck. G. palpalis, G. fuscipes, and G. tachinoids all feed on wild and domestic suidae, bovidae, waterside birds, varanus, and crocodiles. G. pallidipes and G. longipalpus primarily feed on bushbucks. G. longipennis feeds on rhinoceros, bovidae, elephants, and ostriches. And G. M. submorsitans feeds on various birds excluding ostriches. Waterbuck are not targeted by Glossina due to their repellent volatiles, which are being tested as livestock repellents. The genome of Glossina morsitans, a species of tsetse fly, was sequenced in 2014. Tsetse flies have 3 bacterial symbionts: Wigglesworthia, Sodalis, and Wolbachia. Wigglesworthia is the primary symbiont found in the fly's bacteriocytes, while Sodalis can be found either intercellularly or intracellularly. The third symbiont is a type of Wolbachia. The salivary gland hypertrophy virus leads to abnormal bleeding in the crop lobes of GM centralis and GM morsitans. Tsetse flies are blood-feeding parasites that belong to the order Diptera and the superfamily Hippoboscoidea. They are part of the Glossinidae family, which is one of 4 families of blood-feeding obligate parasites. There are up to 34 recognized species and subspecies of tsetse flies, all classified under the genus Glossina. The Glossinidae family is typically placed within the superfamily Hippoboscoidea, which includes other families of blood-feeding insects. The Tsetse genus is divided into 3 groups of species based on their distribution, behavior, molecular characteristics, and physical appearance. The genus includes various species. Fossil glossinids have been found in the Florisun Formation in North America and the N-Spell Lagerstadt in Germany. These fossils date back to the late Eocene and late Oligocene periods. Glossina, also known as tsetse flies, are mainly found in grassland and forested regions of the Afrotropics. However, there are two subspecies, G.F. fuscipes and G.M. submorsitans, that can be found in the southwestern part of Saudi Arabia. In the past, G. tactinoides was reported near Aden in southern Yemen, but there have been no recent confirmations of its presence. Tsetse flies are biological vectors of trypanosomes, which are single-celled parasites that cause diseases like sleeping sickness in humans and nagana in animals. Tsetse flies acquire trypanosomes from infected hosts and transmit them to uninfected animals through feeding. Trypanosomes have a complex life cycle that involves periods in both the tsetse fly and the vertebrate host. Transmission can occur through mechanical means, where the infected blood is regurgitated into an uninfected host, or through biological means, where the trypanosomes undergo several generations within the tsetse fly before being transmitted. The relative importance of these two modes of transmission is not fully understood. The tsetse-vectored trypanosomiases are diseases caused by different trypanosome species and affect various vertebrate species, including humans, livestock, and wild animals. The diseases have different distributions across Africa and are transmitted by different species of tsetse flies. Human African trypanosomiasis, also known as sleeping sickness, is a deadly disease caused by the parasite Trypanosoma brucei. It is transmitted through tsetse fly bites and progresses from the subcutaneous tissue to the lymphatic system, bloodstream, and eventually the central nervous system. There are three subspecies of Trypanosoma brucei, with Trypanosoma brucei gambiense causing a slower onset of the disease, and Trypanosoma brucei rhodesiense causing a more rapid and virulent onset. Another form of trypanosomiasis, Chagas disease, is caused by Trypanosoma cruzi and transmitted by insects in South America. Animal trypanosomiasis, also known as nagana or sura, is a disease caused by trypanosome species that affects farm animals. It reduces growth rate, milk productivity, and strength, often leading to death. Some cattle species are trypanotolerant, meaning they can survive and grow despite being infected but with lower productivity. Trypanosoma congolense and Trypanosoma vivax are the main species infecting bovine cattle in sub-Saharan Africa, while Trypanosoma simii affects swine. Tsetse flies are the main vector, mostly found in Central Africa. Trypanosomiasis hinders livestock development in tsetse fly-infested areas, with the Ndama breed being the most resistant. The conquest of sleeping sickness and nagana in sub-Saharan Africa would greatly benefit rural development, poverty alleviation, and food security. Managing the diseases involves controlling the vector and directly targeting the disease through surveillance and treatments. Economic analysis shows that the cost of eliminating tsetse vectors is outweighed by the benefits of having tsetse-free areas. Vector control strategies can aim for continuous suppression or eradication, but eradication programs are complex and require the integration of various control tactics. Area-wide interventions are more efficient and profitable when large areas with high cattle populations are covered. Various techniques have been developed to reduce tsetse populations, with older, less effective methods being replaced by newer, more efficient, and environmentally friendly approaches. These newer techniques are not only cheaper but also more targeted in their approach, resulting in better control of tsetse populations. One method used to control tsetse flies involved eliminating their food source by killing the wild animals they fed on. An example of this was the eradication of feral pigs on the island of Principe in the 1930s, which led to the disappearance of tsetse flies. Although the flies returned in the 1950s, the new population was not carrying the disease. Early on, a technique called complete removal of brush and woody vegetation was used to make an area inhospitable to tsetse flies. However, this method was not widely adopted and has been abandoned. The process was initially done by hand, but later a more efficient technique called chain clearing was introduced, which involved dragging a chain between two heavy vehicles. However, continuous clearing efforts were required to prevent regrowth, making it expensive and only practical in areas with large human populations. Additionally, the clearing of woody vegetation is now seen as an environmental issue rather than a benefit. Pesticides have been used to control tsetse flies for many years. Initially, inorganic metal-based pesticides were used in localized efforts. After World War II, organochlorine pesticides like DDT were used in large-scale campaigns. These pesticides were applied as aerosol sprays. Later, more targeted techniques were developed, such as using pour-on formulations of advanced organic pesticides directly on cattle. Tsetse populations can be monitored and controlled using inexpensive traps that attract the flies with electric blue cloth. The traps either collect the flies or expose them to insecticide. Early traps mimic the form of cattle, as tsetse are attracted to large dark colors. Chemical attractants like carbon dioxide have been studied but are not economically viable. A cheaper approach is to use cattle urine. In Ethiopia, traps are used as part of a sustainable agriculture and rural development program. The sterile insect technique, SIT, is a method of pest control that utilizes radiation to sterilize male flies. These sterile males are subsequently released into tsetse-infested regions, where they mate with wild females, leading to no progeny. This technique has the potential to eliminate populations of wild flies over time. SIT is an environmentally friendly control tactic and is typically employed as the concluding element of an integrated campaign. It has demonstrated effectiveness in managing populations of other fly species. The eradication of tsetse flies through SIT can yield substantial economic advantages for rural farmers. The tsetse fly has been associated with challenges in early state formation in regions where it is widespread. In 2012, a study utilized population growth models, physiological data, and ethnographic data to analyze the impacts of the fly. The study discovered that the tsetse fly devastated livestock numbers, resulting in a dependence on slave labor for agriculture and hindering the utilization of organic fertilizers. These consequences had lasting effects on population density and cooperation among communities. Some contend that the tsetse fly exerted a substantial influence on African development, while others maintain a more skeptical view. In the 19th century, Africa experienced depopulation and landscape transformation due to the spread of disease. Specifically, rinderpest and the tsetse fly were the main culprits. Rinderpest, which originated in Asia, caused massive devastation among cattle populations, leading to widespread famine and death among humans. This unfortunate situation created an opportunity for colonial powers to assert their control over the region. Additionally, the tsetse fly, attracted to the newly overgrown bush, further impacted the area by spreading sleeping sickness. As a result, the landscape appeared wild and untouched, which eventually led to the establishment of nature reserves and national parks. However, in the 1990s, the disease resurfaced due to inadequate surveillance and management. Tsetse flies are a significant contributor to rural poverty in sub-Saharan Africa. They hinder mixed farming by spreading nagana, a disease that debilitates and kills livestock. This leads to lower milk and meat output, heightened abortion rates, and diminished cattle worth. Additionally, the tsetse fly transmits African trypanosomiasis, commonly known as sleeping sickness, to humans. This illness affects a substantial number of individuals, ranging from 60 to 70 million, and bears a considerable economic burden. Eliminating tsetse flies would enable enhanced food production and foster economic growth in affected regions. CFM Swinnerton was a significant figure in East Africa during the early 20th century. He conducted extensive research on tsetse ecology, which was crucial for understanding the disease transmission in the region. As a tribute to his work, E. Austin named a taxon G. swinnertoni after him in 1922. Tsetse flies have various immune defenses that make them resistant to trypanosome infection. One of these defenses is the production of hydrogen peroxide, which damages the DNA of the parasites. This limits the number of infected flies in the population. The first source is an article titled Tsetse by George Gerster, published in National Geographic in December 1986. The article provides information about tsetse flies. The second source is a research paper titled Tsetse Genetics, Contributions to Biology, Systematics, and Control of Tsetse Flies by R.H. Gooding and E.S. Kraftsur, published in the Annual Review of Entomology in 2005. The paper discusses the genetic aspects of tsetse flies and their implications for biology, systematics, and control. These are a list of books that provide information on the biology, ecology, distribution, and control of tsetse flies and the trypanosomiases. They cover topics such as the natural history of tsetse flies, the role of trypanosomiases in African ecology, the distribution and abundance of tsetse flies, tsetse biology and ecology, and the struggle against tsetse flies in Africa. In conclusion, the word tsetse has evolved to be commonly used without the term fly in English, particularly in scientific and development contexts. These flies have been extensively studied by entomologists for their medical, veterinary, and economic importance.
Thank you for watching this AGNDD Intellireport. Like, subscribe, and click the bell button to get notified during new uploads. And make sure to tune in next time. Stay safe and healthy. Cheers.